perhaps in 1970 on acetate and Chincoteague.
Oh, really?
Just on the edge of the coast.
Okay, all right. So good morning. I'm Darcy Gentleman, and welcome to this news briefing from the 254th National Meeting and Exposition of the American Chemical Society here in Washington, D.C. We're joined today by Dr. Richard Van Breemen from the University of Illinois at Chicago. He has been studying licorice, which some women now take to treat menopause, and its potential interactions with medications. Dr. Van Breemen.
Thank you very much, Darcy. I direct a botanical center funded by the National Institutes of Health, Office of Dietary Supplements, and the National Center for Complementary and Integrative Health. And our mission has been, since our founding in 1999, to look at botanical dietary supplements with respect to their safety and efficacy to protect human health and to benefit human health. We've been focused on women's health in particular, and those botanicals that women are using as alternatives to hormone therapy at menopause. This became particularly critical in the early 2000s with the first publications from the Women's Health Initiative that indicated that hormone replacement at menopause offered certain benefits, such as control of menopausal hot flashes and osteoporosis, but didn't deliver on the promise of preventing dementia, degenerative neurological diseases, or heart disease. Hence, many women started turning to alternatives. Also, they were concerned about the risk of estrogen therapy promoting certain hormone-dependent cancers like breast cancer. So women are hoping that botanical dietary supplements as alternatives to conventional hormone therapy might help them control menopausal symptoms like hot flashes, but at the same time not contribute to risks like breast cancer and dementia and heart disease. So licorice is one of those botanicals that is marketed for the control of menopausal hot flashes. Licorice does contain estrogenic compounds. It contains cancer chemopreventive agents as well. And our interest has been to look at the efficacious effects of these botanicals, their potential risks, and hopefully identify specific compounds that could benefit women and control or diminish those in products that might be detrimental to their health. There are multiple species of licorice that have been used for many generations in the history of human health and medicine. 3 are particularly used in current botanical supplements available in the United States and elsewhere. 2 of them are Asian species, which have a long history of use in traditional Chinese medicine, and one of them is a European Mediterranean species, which we all know in the form of candy and a flavoring agent for confections of many kinds. Both the European and the Asian species have had a long history of use in various medicines, not just traditional Chinese medicines for the Asian species, but Western medicines, because licorice has a sweetness, a compound called glycyrrhizin, which enhances the sweetness of licorice. It's about 50 times more potent than sucrose, conventional table sugar. And as we all know, licorice has certain anise-like flavoring agents as well. But also contained in licorice are compounds that have chemoprevention properties, like licochalcone A and the chalcone isoliquertigenin. But they're estrogenic compounds like liquertigenin that can have promise for controlling menopausal symptoms. So our aim at the Botanical Center that I direct in Chicago is to develop a product which has a balance of both estrogenic compounds to help women manage menopausal symptoms, but at the same time be rich in chemoprevention agents that can help prevent perhaps estrogen-related cancers, dementia, heart disease, and other, say, problems that can be associated with oxidative stress and inflammation. Not all species of licorice are the same. There are some unique compounds. There are compounds that are shared amongst all the species. So we believe that all these products that are marketed should not only be labeled with respect to which species these products contain, but hopefully be standardized. And we've advocated for standardization of botanical supplements, both chemically and biologically, through bioassays to make them biologically understood, but chemically, so that we can determine what the levels of specific compounds are that might have estrogenic effects or chemopreventive effects. And then consumers will have the ability to choose those products that will suit them the best from the marketplace. Now, with respect to the safety of botanical dietary supplements, and licorice in particular, we're concerned about a little understood and little explored thus far potential for these products to interact with drugs and prescription drugs in particular. About half of all Americans report using botanical dietary supplements. About 15 to 20% also report simultaneously using prescription medicines. If these botanical dietary supplements can interfere or potentiate with the effects of these prescription medicines, that could be a cause for concern. There are certain botanicals that are well documented to interfere with what we call the pharmacokinetics of medicines and prescription medicines. The best documented is St. John's wort, which induces an enzyme that's responsible for about 2/3 of all drug metabolism, called cytochrome P453A4. By inducing this enzyme, all prescription medicines that are metabolized, usually that's the degradative pathway leading to the elimination of compounds from the body, induction of this enzyme can lead to these medicines being metabolized and eliminated faster than before. So any compounds or any drugs that are metabolized by this particular enzyme would have a shorter half-life, a shorter efficacy, and be less efficacious. On the other hand, botanicals like golden seal have the opposite effect, and then these can induce, or rather than induce, can inhibit the metabolism and the activity of drug metabolizing enzymes. And as a result, drugs that are metabolized through that pathway will have longer half-lives, have greater effect, and potentially toxic side effects as a result. So with respect to licorice, there are species that can inhibit and induce. We're finding that certain licorice species can either induce or inhibit certain drug metabolizing enzymes. They're not all the same. One needs to pay close attention to which product contains which of these species. We're working very hard to define and identify specific compounds within each of the licorice species that are responsible for these induction or inhibition effects. Currently, we're finishing our in vitro work. I'll report today on our in vitro data, and we're planning a clinical trial beginning next summer to test whether what we're learning in our laboratory-based studies have these same effects in the clinic. Thank you.
All right, thank you very much. Are there any questions? And I'll remind those to please state your name and affiliation before asking your question.
Hello, Ben Valsler from Chemistry World magazine. Is there any solid literature out there from clinical trials to show that licorice is effective at preventing hot flashes and things like that? And with that in mind, how do you think, what are you going to need to show in your clinical trials to sort of cancel that out and balance that out?
There have been a very small number of trials about efficacy of licorice species in controlling hot flashes. There's a wealth of information about licorice being useful for sore throats, to help fight certain viral infections or bacterial infections. There's actually very little information currently in the literature about the efficacy of licorice products in controlling menopausal symptoms like hot flashes. That is certainly one of the goals that we're working towards. We want to establish a product which is standardized before we go to the clinic, and we want to make certain that it contains the correct species, that it's botanically authenticated, and we want to then be able to carry out a controlled clinical study to measure efficacy. But we're not quite at that point yet.
Do you think, therefore, it's irresponsible to market licorice as a supplement for women going through the menopause?
Well, products like licorice, botanical supplements, have many generations of human experience. And they certainly should be regarded at this point in history of medicine as most likely safe as used in practice historically. And if you read the ethnobotany on how these products are used, I think licorice products are certainly safe. There's the chance that one could prepare a product that's extracted in such a way as to concentrate certain compounds so that they wouldn't be safe. There is a compound, as I mentioned, the sweet component of licorice, glycyrrhizin, which is metabolized to an acid which has some known toxicity or side effects. It works in the kidney to inhibit enzymes related to the formation of aldosterone. So hyperaldosteronism is a side effect of using licorice products that contain too much glycyrrhizin. So that's actually controlled right now, and levels of glycyrrhizin are typically not very high in licorice products that are currently in use. With respect to the efficacy, that's another question, and that we'll have to wait for placebo-controlled, randomized clinical trials to prove that.
And just finally, are there any particular drugs that you're worried about the drug interactions, perhaps ones that are particularly common in the demographic of women going through the menopause?
Despite the fact that there have been quite a few of laboratory-based in vitro studies of drug-botanical interactions, very few of these thus far has proven to be a problem in the clinic. I mentioned two in particular, golden seal and St. John's wort, that have been clinically proven to have drug interactions. But fortunately, most of the preclinical modeling hasn't led to clinically relevant or significant effects in terms of drug interactions. And I hope that will certainly be the case in licorice, which we'll be investigating soon in the clinic. We're also working towards improving the predictive models that we use in our laboratory-based studies, and we're moving towards using physiologically based pharmacokinetics modeling, PBPK for short, which we hope will improve the predictability of our in vitro testing.
Thank you very much.
Thank you. Other questions?
Doug Dollimore, American Chemical Society. Doctor, based on your findings, what advice would you give to women about licorice? Should they consult their doctor? Should they just stop taking it until this whole labeling is clarified?
Well, we've advocated for a stepwise process by which botanical dietary supplements are obtained, extracted, prepared, standardized, tested, both in preclinical models and then in the clinic for efficacy and safety. One of the steps in this process that we've advocated since we started our work in 1999 is for all of the botanical dietary supplements marketed to be prepared using good manufacturing practice. And that is now enforced by the FDA. And we consider that one of the steps in the progress of this field that we've always believed in, and now the FDA is also taking that particular approach. I think more than anything else, good manufacturing practice has led to a safer product of all the products on the market in the U.S. that follow this approach are safer than ever before. We hope that eventually more of these products will undergo clinical testing of safety and efficacy as well. Until that happens, I would advise women to certainly tell their health care provider, their clinician, their doctor, their nurse, tell these practitioners that they are using dietary supplements, because then prescription medicines that they might take thereafter or concomitantly can be used more safely.
All right. We have a question from online, I believe. Oh.
Actually, this is from me, Katie Cottingham, American Chemical Society. What will you do next with this project?
We are currently carrying out clinical studies of safety with respect to drug-botanical interaction on hops. That will finish by September. Next month, we're beginning another clinical trial on red clover, another botanical used by women as an alternative to hormone therapy. And next summer, we're going to begin a study on licorice interactions. So the licorice is, for next summer, our next step in our drug botanical work. Longer term, we're hoping to carry out studies of efficacy, and that requires more funding than we currently have.
Okay, and is your end goal to make a product eventually of this licorice, the extracts?
We certainly are not opposed to the products that we develop being marketed. That is not our current goal, and we don't have any collaborations. I have nothing to declare with respect to conflicts of interest there. We have no partners with whom we've promised to develop any products, but we hope that if we do develop a product in an extract that we can demonstrate to be safe and efficacious, we would welcome the opportunity to market that.
Christine Sa, American Chemical Society. So you mentioned that the licorice has estrogenic compounds in it, and I am assuming that's the active, bioactive part that's potentially treating the menopause symptoms. Are you also looking at mechanism? How do these compounds act differently and enact their effects versus hormone replacement therapy?
Certainly. Mechanism of action is one of the steps in our process. We have an 8-step process to develop safe and effective botanical dietary supplements. Looking for mechanisms of action and identifying the compounds responsible as part of that process. We use a battery of assays in our laboratories to look for estrogenicity, which could be through binding to the estrogen receptors alpha or beta. It includes cell-based assays and some rodent-based assays. So once we find effects of our extracts, we'll subfractionate those and isolate and identify the specific compounds with estrogenic activity. We want to know if those estrogenic compounds target the ER alpha or the estrogen receptor beta. It is preferable to have compounds that are more selective for ER beta than alpha, and it has to do with the safety profile. Estradiol, the endogenous estrogen that's most potent, targets both ER alpha and beta. But those effects on ER alpha tend to promote proliferative activity of cells, which can help promote breast cancer and other hormone-dependent cancers like uterine cancer. And so we're trying to find a botanical alternative which is more estrogen receptor beta selective, which should have a safer profile than estradiol itself.
Thank you.
Okay. Question here in the room.
Bela Buslik, ACS Communication. You've discussed about 3 different species in your abstract, and
yes.
How would you standardize something like this, where a couple of them have more compounds than others, and glycyrrhizin apparently is the main component of each one of them, but some affect the cytochromes more than others. So how would you go to so the customer actually knows what they're buying?
The FDA guidelines for good manufacturing practice require botanical authentication. That means that by law, a licorice product sold as a botanical dietary supplement must specify what species it contains. In our testing of the marketplace, some of these products are labeled accurately. Some don't actually say which species of licorice, they simply say licorice. We believe that the specific species should be indicated. In our testing, there are compounds that are unique to one species or another. For example, licochalcone, a chalcone with chemoprevention activity, is found only in Glycyrrhiza inflata. That's one of the Asian species of licorice. On the other hand, there's a compound globardin that's only in the European Glycyrrhiza glabra species. Some of the compounds that we've identified as causing or potentially causing drug botanical interactions, such as the induction and others inhibit cytochrome P450 enzymes like cytochrome P453A4, are found only in one species and not always in all 3. So I believe that a product could be developed which contains one species in particular, and that it's optimized to have a good balance of estrogenic and possibly anti-estrogenic properties, as well as chemoprevention activity.
Okay, how would the customer actually distinguish between licorice? People know licorice, it tastes like licorice. It's like several other vegetable species that have the same compounds in them. They're very, very different. When it's called licorice root, let's say, what does one do to protect themselves against the ones that perhaps would be more interactive than the other?
Well, I hope the answer to that is good research. And I'm hoping one day that products on the market will actually have labels that indicate maybe references to and maybe package inserts that point to specific good quality research studies that support safety and efficacy of that particular product. And then a consumer should be able to judge, do they want a licorice product that has got a balance of natural products that seem to be beneficial for perimenopausal women, or a different one that might be best for postmenopausal women, or even a product that might be better because it's rich in chemoprevention agents for younger women.
Thank you.
All right. If there are no further questions, all right. Thank you very much, Dr. Van Breemen, and all of you for coming. The archived version of this session will soon be posted at bit.ly/ACS live_DC. That's capital A, capital C, capital S, capital L, lowercase IVE _ capital D, capital C. Please join us for our next press conference at 1 p.m. today on tapping elite athletes' microbiomes to give the rest of us a fitness edge. Thank you very much.
Thank you.